In 2010, Svante Paabo's team sequenced the Neanderthal genome and proved what decades of consensus had denied: Homo sapiens and Neanderthals, interbred. Every non-African alive carries 1 to 4% Neanderthal DNA. Collectively, 40% of the Neanderthal genome survives across living humans. That should have settled the question. It didn't. The same DNA that proved interbreeding exposed a pattern scientists still cannot fully explain. The X chromosome is 5 times more depleted of Neanderthal ancestry than the rest of the genome. Something was purging it. The reason reaches the deepest unresolved problem in human evolution. Svante Paabo, director at the Max Planck Institute for Evolutionary Anthropology, led the team that sequenced the first high-coverage Neanderthal genome from Vindija Cave in Croatia. Their 2010 paper in Nature, with Richard Green as first author, applied next-generation sequencing technology to extract and assemble ancient DNA from 3 Neanderthal bones. The resulting genome exceeded 1.3 billion base pairs in coverage, a technical milestone for paleogenomics. By comparing these sequences to modern human reference genomes, the team identified thousands of single nucleotide differences and mapped patterns of shared ancestry. Their analysis showed that non-African populations carry between 1 and 4% Neanderthal DNA, a figure calculated using haplotype sharing and D statistics. The data revealed that all non-African lineages, Europeans, East Asians, South Asians, and Indigenous Americans, share the same admixture signal pointing to a single interbreeding event after modern humans left Africa, but before they dispersed across Eurasia. The paper quantified the Neanderthal contribution as a genome-wide average, with no evidence for recent or ongoing gene flow. The conclusion was clear. Neanderthal DNA is a permanent feature of the modern human genome outside Africa. The publication triggered immediate global debate, as the 1 to 4% statistic forced researchers to re-examine the boundaries between species, the definition of hybrid viability, and the evolutionary consequences of archaic admixture. Paabo's team set a new standard for ancient DNA research and opened the door to genome-wide mapping of Neanderthal fragments in living people. David Reich's group at Harvard led the effort to map Neanderthal-derived fragments across the full spectrum of modern human genomes. Their analysis, published in Nature, pooled data from over 2,500 individuals in the 1,000 Genomes Project. Each non-African individual carries about 1 to 2% Neanderthal DNA, but the specific segments differ from person to person. When all surviving Neanderthal fragments are aggregated across global populations, approximately 40% of the Neanderthal reference genome is represented somewhere in living humans. This cumulative figure does not mean any one person carries 40%. It reflects the collective, non-overlapping coverage of Neanderthal sequence in the modern gene pool. The pattern is mosaic. Some regions of the Neanderthal genome are present in many people, others are found in only a few, and some are missing entirely. The largest coverage comes from populations in Europe, East Asia, and South Asia, each contributing distinct sets of fragments. In contrast, sub-Saharan African populations show less than 0.5% Neanderthal ancestry, a signal attributed to back migration from Eurasia after the initial admixture event. These Neanderthal-derived tracks in Africans are shorter and less frequent, consistent with introduction through secondary gene flow rather than direct contact. Reich's modeling quantifies this backflow using haplotype length distributions and population structure analysis, confirming that the majority of Neanderthal DNA outside Africa traces to a single pulse of admixture around 45,000 to 49,000 years ago. The resulting global map reveals both the reach and the limits of Neanderthal introgression, fragmentary, uneven, and shaped by subsequent human migrations. This atlas of surviving Neanderthal DNA sets the foundation for dating admixture events and for investigating why certain genomic regions reject archaic ancestry. Alper Sümer and colleagues published a high-coverage analysis of early modern human genomes from Ranis, Germany, and Zlatý Kun, Czechia, in Nature in 2025. Their team applied segment-length decay models to these genomes, using the DATES algorithm to convert the distribution of Neanderthal-derived DNA tracks into a precise estimate of the admixture event. The mean Neanderthal fragment length in these individuals is 0.42 centimorgans, corresponding to roughly 1,250 generations before death. Radiocarbon dating places the Ranis burial at 45,200 years before present, and the Zlatý Kun tooth at 46,300 years, both with margins of error below 400 years. Stratigraphic and archaeological evidence from these sites matches the genomic clock, supporting a narrow admixture window between 45,000 and 49,000 years ago. The tract length method relies on the principle that after admixture, recombination breaks down long DNA segments from one population into shorter fragments every generation. By measuring the rate of this decay in ancient genomes, Sümer's group could pinpoint the timing of gene flow with greater resolution than previous studies. The results show that all non-African populations share this admixture pulse, with no evidence for substantial later Neanderthal input. This timing aligns with the last robust Neanderthal presence in Europe and the initial expansion of modern humans through the Dinaric Balkan corridor. The 45,000 to 49,000 year date range now serves as the temporal anchor for selection analyses, demographic modeling, and the investigation of reproductive barriers. Sümer's findings close the uncertainty around when Neanderthal DNA entered the modern human gene pool and set the stage for examining how these archaic fragments have been retained or purged over time. Genome-wide scans reveal a pronounced depletion of Neanderthal DNA on the human X chromosome. Sankararaman and colleagues, publishing in Nature in 2014, measured Neanderthal ancestry on the X at just 0.5%, compared to 2% on the autosomes, a five-fold reduction. This differential persists after correcting for lower SNP density, indicating a true biological effect rather than a technical artifact. The same studies found that regions of the genome with high testis-specific gene expression contain even less Neanderthal sequence, with some loci showing a 70% depletion relative to the genomic background. These patterns are consistent with Haldane's rule, which predicts that hybrid male fertility is more likely to be compromised when two lineages are diverging toward reproductive isolation. In this context, the heterogametic sex, human males who carry only one X chromosome, would be more vulnerable to incompatibilities introduced by Neanderthal alleles. Simulations from Reich's group at Harvard and from Kelly Harris at the University of Washington support this interpretation, showing that a modest selection coefficient, S equals 0.001 to 0.004 per locus, is sufficient to explain the observed X to autosome deficit. Forward modeling demonstrates that selection against Neanderthal variants in genes expressed in the testes can drive a multifold reduction in just a few hundred generations. The X to autosome depletion ratio, calculated as 0.5% divided by 2%, yields 0.25, matching the theoretical expectation for hybrid incompatibility under Haldane's rule. These findings point to a functional barrier at the level of male fertility and suggest that, while interbreeding occurred, the genetic legacy of Neanderthals was systematically purged from regions critical to reproduction. This chromosome-level evidence sets the stage for modeling the strength and timing of selection acting on archaic DNA after admixture. Genome-wide analyses after the initial admixture event show a steep drop in Neanderthal ancestry, from an estimated 10% in early hybrids to about 2% in present-day non-Africans. Sankararaman and colleagues, in their Nature paper from 2014, modeled this decline with a selection coefficient S between 0.001 and 0.004 per locus. This value is sufficient to explain the rapid loss of Neanderthal DNA over roughly 1,500 generations. The strongest purging is observed on the X chromosome and in genes with high testis-specific expression, but the effect extends across the genome. Large-scale mapping projects, including those led by David Reich at Harvard, identified extensive regions nearly devoid of Neanderthal sequence, known as introgression deserts. The most prominent desert spans 17 megabases on chromosome 7, encompassing the FOXP2 gene, a transcription factor linked to speech and language. In this region, Neanderthal DNA is virtually absent in all surveyed modern genomes. The FOXP2 desert is not an isolated case. Additional deserts occur on chromosome 8p23.1 and in loci involved in male fertility and immune function. These patterns are not artifacts of sequencing or analysis. Multiple independent methods, including conditional random field models and hidden Markov models, converge on the same regions of depletion. The scale and consistency of these deserts indicate that purifying selection systematically eliminated Neanderthal alleles from functionally sensitive regions. Selection modeling shows that even a modest fitness cost per introgressed allele can account for the observed genome-wide reduction. The empirical data, including percentages, tract lengths, and the spatial distribution of deserts, support a scenario in which hybridization was initially successful, but followed by strong selection against incompatible variants. The FOXP2 region, with its complete absence of Neanderthal sequence, stands as the most extreme example of this selective purge. Genomic clock analyses place the split between Neanderthals and modern humans at roughly 400,000 years ago. Prüfer and colleagues, using high-coverage Neanderthal genomes, calculated the most recent common ancestor at 415,000 to 447,000 years before present. Rogers and collaborators refined this estimate to a range of 380,000 to 440,000 years using joint site frequency models. Chris Stringer of the Natural History Museum in London points to this deep divergence as the anchor for species separation. He argues that the accumulated differences in cranial shape, dental patterns, and adaptive alleles meet the criteria for distinct species under both biological and phylogenetic definitions. Meneganson, working from morphological and ecological data, supports this view, emphasizing that a prolonged period of allometry allowed Neanderthals and Homo sapiens to develop unique adaptive profiles. According to this model, the later admixture event was a secondary contact between separate species, not evidence of ongoing conspecificity. Svante Paabo, director at the Max Planck Institute for Evolutionary Anthropology, challenges this position. He notes that the same genetic record reveals persistent gene flow, shared polymorphisms, and the survival of Neanderthal haplotypes in present-day genomes. Paabo favors a metapopulation framework where intermittent interbreeding and incomplete reproductive isolation prevent a clean species boundary. He cites the Denisova 11 genome, a first-generation hybrid of Neanderthal and Denisovan parents, as proof that archaic hominins exchanged genes despite long divergence times. Paabo's model rejects strict taxonomic separation and instead describes a species complex with porous boundaries. The debate remains unresolved. Stringer and Meneganson point to the 400,000-year split and the evidence for hybrid incompatibility, such as X chromosome depletion and purging of Neanderthal DNA from key reproductive regions. Paabo and his colleagues argue that the persistence of archaic alleles and the existence of fertile hybrids undermine any hard boundary. Both camps use the same genomic data to support opposing taxonomies. The question of whether Neanderthals and modern humans were separate species or diverging populations within a single metapopulation remains open, shaped as much by definitions as by DNA. The Neanderthal O blood type allele, identified in 2 L-sidron individuals by Lalueza Fox's group, matches the modern O01 haplotype found in about 45% of people worldwide. This allele, a loss-of-function variant in the FUT1 gene, appears at similar frequencies in sub-Saharan Africans, Europeans, and East Asians. Genome-wide analyses estimate that roughly 12% of O-type haplotypes in non-African populations derive from Neanderthal introgression. Denisovan genomes also carry O alleles, but their mutations differ, indicating independent origins. Functional studies link the O allele to a reduced risk of severe malaria and certain bacterial infections, suggesting a selective advantage in pathogen-rich environments. Neanderthal-derived pigmentation alleles also persist. The MC1R variant P ARG151 cis, present in Vindia and L-sidron Neanderthals, is associated with red hair and fair skin in modern Europeans, where it occurs at about 4% frequency. At the OCA2HERC2 locus, a Neanderthal-derived blue eye allele appears in roughly 3% of Eurasian genomes. The BNC2 gene carries a Neanderthal enhancer linked to lighter skin, now present in about 15% of Europeans. These archaic variants demonstrate that some Neanderthal DNA was retained because it provided adaptive benefits in new environments. The Levant corridor served as a critical meeting ground for Neanderthals and early modern humans between 120,000 and 40,000 years ago. Archaeological remains from sites such as Amud in Israel, with a Neanderthal brain volume measured at 1,740 cubic centimeters, reveal individuals with both robust Neanderthal traits and features approaching those of Homo sapiens. Burial evidence from La Chapelle-aux-Saints in France, dated to approximately 65,000 years ago, documents deliberate interment and ochre use, indicating symbolic behavior. At Krapina in Croatia, over 20 eagle talons, pierced and worn as ornaments, have been dated by uranium series analysis to around 130,000 years ago. These artifacts predate the earliest Homo sapiens beadwork and demonstrate abstract thinking and social signaling among Neanderthals. The paleo environment of the Levant fluctuated between Mediterranean woodland and steppe, creating seasonal corridors that facilitated repeated contact. This ecological and cultural context grounds the genetic evidence of admixture, and highlights that Neanderthal-modern human interactions unfolded in landscapes rich with symbolic practice and behavioral complexity. Introgressed. Neanderthal DNA continues to shape health outcomes in living people. Genome-wide association studies have linked archaic variants to immune response, disease risk, and drug metabolism, toll-like receptor haplotypes inherited from Neanderthals, including variants in TLR1, TLR6, and TLR10, increased sensitivity to bacterial and fungal infections, but also heightened risk for allergies and autoimmune conditions. A Neanderthal-derived haplotype on chromosome 3, identified by Zeeberg and Paabo, doubles the risk of severe respiratory illness and COVID-19. Other introgressed alleles in the OAS gene cluster modulate antiviral activity, influencing susceptibility to viral hepatitis and West Nile virus. Pharmacogenomic studies reveal that Neanderthal variants in genes such as CYP2C9 and CYP2C19 affect how individuals metabolize common drugs, altering the efficacy and toxicity profile of anticoagulants and antidepressants. The clinical impact is uneven, and some archaic alleles confer resistance to pathogens, while others increase vulnerability to chronic inflammatory diseases. These associations are strongest in non-African populations, which retain 1 to 2% Neanderthal ancestry on average. The ongoing presence of archaic haplotypes in immune-related loci demonstrates that the genetic legacy of Neanderthal admixture remains clinically relevant, shaping disease outcomes and drug responses in the present day. Today, fragments of Neanderthal DNA persist in every non-African genome, yet genes on the X chromosome and those active in testes are almost entirely purged. This paradox, documented by Paabo's Max Planck team and published in Science and Nature, underscores that reproductive barriers emerged even as gene flow occurred. The line between species and population remains unresolved, demanding sharper definitions as new ancient genomes challenge every assumption. What boundary will we draw next?